Hello everyone, welcome back to the Green Chemistry Education webinar series hosted by the Green Chemistry Commitment. My name is Amy Cannon. I'm the Executive Director of Beyond Benign, a nonprofit organization dedicated to green chemistry education in K-12 through higher ed. This webinar series is designed to highlight relevant topics in green chemistry education for faculty and students. So as a reminder, we're broadcasting live and recording this session, so all attendees are in listen-only mode and all lines are muted. So if you have a question, you can type it in anytime in the question chat box there on your control panel. So feel free to type it right in there at any time. We're going to respond to as many questions as we can after the presentation by our two speakers today. The recording will be posted on the link that you see in the window chat box and also on this slide here. And any supporting documents such as presentation slides are also posted on that link as well. And there on that link you'll also find our webinar archive. So we have a whole archive of past webinar recordings and supporting documents that you're able to access there as well. For those of you who participate in social media, please connect with us on Twitter or Facebook. And as always, we'd love to hear some conversation going on about this specific webinar or about green chemistry in general. So feel free to join in. And thanks so much for joining us here. This webinar is being brought to you as part of the Green Chemistry Commitment Program, which is a consortium program aimed at transforming chemistry education, expanding the community of green chemistry practitioners, growing departmental resources for those looking to adopt green chemistry, improving connections to industry, and affecting systemic change in chemistry education. The program is voluntary, flexible program for adopting green chemistry student learning objectives and for promoting the work that you're already doing at your institution. You can find more information on our website link here or email me or our new program manager, Derek Ward, directly. As part of our webinar series, we're giving away a green chemistry book at the conclusion of each webinar, and I seem to have lost one of my resources on my slide here. We do have a toxicology handbook that we have available specifically that we got in for this webinar. So we'll be announcing a randomly selected winner from our list of attendees at the end of the webinar, and we'll send that book right out to you. So look for that announcement at the end. Okay, with that, we are very happy to have Dr. Pamela Spencer and Dr. Craig Rowlands join us for a discussion on toxicological tools for greener product design. So I'm going to briefly introduce both of them, and then I'm going to hand the controls over so we can hear directly from them. So we're very excited to have two toxicologists with us today and share their experience and their work with us. Dr. Craig Rowlands is a senior scientist in supply chain and sustainability at Underwriters Laboratory. He's an industry leader with extensive experience at Dow Chemical and beyond as a regulatory toxicologist. He received his PhD in toxicology from Texas A&M University, and he is also a diplomat of the American Board of Toxicology. Dr. Pamela Spencer is the Senior Director of Regulatory and Product Stewardship at Angus Chemical Company. She also has extensive experience in regulatory toxicology testing as well as research to define human health risks. She holds a BS in biology from Saginaw Valley State University, an MS in toxicology from Central Michigan University, and a PhD in toxicology from the University of Michigan. And she's also a diplomat of the American Board of Toxicology. So we're very happy to have these two join us today, and we're going to start off with Craig, and then we'll go over to Pam. So Craig, I'm going to hand the controls over to you.
Okay.
All right, is that coming up for you?
Nothing yet. Hang on, here we go.
Got it, okay.
And make sure I get the right screen.
I could try to do it again if you think it's, oops.
Okay, I think we're good.
You got it?
Let me know when you can see that.
Okay, it's coming up. Hold on. Sorry, it's taking a bit to load. There it is. Okay, we got it.
Okay. Okay, good. Okay, great. Well, thank you, Amy, for the opportunity to present to this organization and this particular topic. Again, my name is Craig Rowlands. I'm a senior scientist at Underwriters Laboratories. And what I want to take you through today in my time is really give you a little bit of background on some of the drivers that we see at Underwriters Laboratories that really indicate that predictive toxicology is the way that the companies need to go when they're dealing with the issue of trying to reduce the hazards in the chemicals they're producing and in the products that they're making. You all work with chemical companies all across the supply chain, from those who produce the individual chemicals to those who formulate them, the retailers, and even end-of-life issues. So we hear a lot from different parts of that supply chain and the drivers that really are driving us at Underwriters Laboratories to get more into predictive toxicology so we can help those that we service in this industry. I'm going to start with just a little bit of background on why green chemistry is really needed, what's driving the decision to move into this, the business case for it. I want to provide you a little bit of background of how toxicology is done and how predictive toxicology works, the actual methodologies and approaches. And then I will leave you with a sense on a couple of different QSAR tools, cheminformatic tools, especially one that we're working with Johns Hopkins University to develop that we're using for some of our customers and also internally for a lot of the things we do in UL. So just kind of a little bit of background, chemists are very busy. We make a lot of chemicals. There are over 130 million chemicals now that have CAS numbers, and predictions are that even though the population of our planet is going to remain fairly stable over the next 30 years, our actual production of chemicals that we're using for all those products and technologies that we love in our everyday lives is really going to double over that same period of time. Only a small fraction, though, of these chemicals are used in industrial purposes. For example, I think one estimate here is 140,000 of those are actually using the products that you use every day, the furniture you're sitting on, the room you're in, the various products you use in your daily lives. In addition, we also come in contact with a lot of chemicals through the personal care products we use. Over 10,000 chemicals are used on a particular shower this morning. I've certainly used a lot of those products. We use multiple different types and multiple different kinds of chemicals that we come in contact. We also consume a lot of chemicals in our diets. Over 4,500 or so are used in food additives. So we get exposed to chemicals every day, multiple types from different sources, but also from different routes. So we inhale them, they come in contact with our skin, we actually ingest them, and we drink them in the beverages that we consume. It's these considerations and this understanding over the last decades that have driven a lot of concern over whether or not chemicals are actually assessed appropriately. Do we know enough about their potential health effects in terms of the human health, but also their impact on the environment and the world that we live in? And so this has led many governments to respond by increasing regulations on the testing and assessment of chemicals for their human health and environmental impacts. This is a pretty dramatic increase. If you look at this curve on the graph here, it's really going up almost exponentially. The big change happened almost a decade ago in Europe when they introduced what's called the REACH registration requirements. And this required that every chemical that's manufactured, imported, marketed in the European Union had to be assessed for human health and environment, and a full dossier of assessments for endpoints for both of the human health and environment had to be included along with risk assessments, some of the higher volume chemicals. This is the last year of those registrations for the first round, May 2018, and when those are fully submitted, all of them, there's going to be over 30,000 chemicals just in this last round alone that will be submitted. One of the largest exports in the regulatory world was actually REACH. Many other countries have picked up on this and are developing their own sort of REACH type registration requirements. And even in the United States, we now have a new chemical law we call the Lautenberg Chemical Safety Act. This is the amended or updated Toxic Substance Control Act. It was a 40-year-old law, but now for the first time in the U.S. for industrial chemicals, there is a requirement for a risk assessment to be conducted and reviewed by the U.S. EPA, and an affirmative finding of no unacceptable risk must be found for that chemical to be marketed in the U.S. For these various industrial uses. And so in addition to all the requirements just for for the intended use of chemicals, there's always new types of concerns that are popping up, such as endocrine disruptors that came up a little over a decade ago. We're always concerned about mixtures. How do we assess those? The complications associated with testing multiple chemicals using conventional toxicology approaches. There's always new technologies and new chemicals. For those technologies, nanomaterials in the last decade or so, we're seeing in our own work at UL, we're starting to work with companies that are developing products for 3D printing. We're seeing that being used across the chemical supply chain for lots of different purposes, and interesting chemistries are being developed to be used for those 3D printing approaches. And so, in addition to what's regulated, though, because many thought that governments weren't moving fast enough, there were voluntary sort of sustainability frameworks that have been created over the last several years. A lot of these are what we call chemical alternative assessments, and these are developed to substitute for the use of hazardous chemicals and products. Again, these are voluntary, but they actually drive a lot of decisions upstream in the supply chain. There's really over 100 of these tools that have been developed for comparing chemical characteristics, different chemicals. This is a very popular one on the screen here. This is called Green Screen. And as you can see across the top row, what it does is it assesses the potential health impacts across multiple endpoints for human health and environmental endpoints. And what one does is uses a fairly simple color scheme and coding scheme where green is low hazard, yellow is moderate hazard, red is high hazard, a gray box or a white box there would be no data, the data is missing. And that information is filled in with hazard information for each one of these chemicals. And green, it's easy to see when you compare these side by side. You can either use multiple chemical ingredients and see what you're looking for is the lowest number of green boxes versus red boxes, the lowest number of L's versus H's. But it requires a lot of toxicology information to be determined and gathered in order to fill these in completely so you have a fair comparison. Nevertheless, these schemes are very popular and are really driving, I said, a lot of decisions in the chemical industry, especially all the way up to producers, whose chemicals are being assessed in these types of frameworks. These are used also by retailers to determine what products they may sell on their store shelves. You know, for example, Walmart has introduced lists of chemicals they don't want to see in the products they sell. Walmart is big enough that when they decide not to sell a chemical in one of their products, that's heard all the way down to the individual chemical producers because they're losing a very large market. In many ways, these decisions by these retailers for these very these voluntary frameworks are driving more changes in the chemical industry than any chemical law that's been made in the last 20 years. So chemical pressure or pressures the chemical companies face are really quite extensive, starting all the way from the producers up through the supply chain, which is very complicated. Those decisions that an ingredient manufacturer makes at the very beginning of the supply chain will affect many downstream decisions and have lots of very costly implications. So green chemistry is the way in order to get these decisions made in a way that you'll minimize the impact or negative impacts down that supply chain. By developing better ingredients, lower hazard ingredients, one can produce better results in alternative assessments, have a better opportunity to get into different products. Your hazard communication decisions will be less consequential. For example, if a chemical that is a highly potent or category 1 toxicant may not be allowed to be shipped in an airplane, would have to be shipped in a truck. So those kinds of decisions have really tangible and practical implications, and that can change the way you market products for your customers. Again, I've already talked about retailers are using different hazard assessment frameworks to determine whether or not they will sell products in their store space based on the hazard profile of chemicals. They also have to comply with many regulations that are driven by chemical hazard profiles. And then key performance indicators and sustainability metrics often include chemical hazard assessments, and those scores will determine how well or how receptive consumers are to one's products. Well, there's lots of different decisions that are made here and many different pressure points on what has to be included in those decisions. One thing that was very obvious to us as we worked all the way across the supply chain with our customers is that just about every one of these has a requirement for hazard data. Chemical hazard data is required to assess each one of these different steps along that supply chain. And so that's really why we decided to get into predictive toxicology, because we needed a way to help our customers meet their requirements and their needs, and we needed to do this in a way that didn't require a lot of effort in terms of animal testing and time. So predictive toxicology was a very good solution for us. So I'll talk a little about what we developed with our colleagues at Johns Hopkins University. But really, I just want to turn my attention now very quickly to why can't we just use conventional toxicology to get the job done? All right, we've been doing this for 50 years. We know how to do conventional toxicology testing, but why is this no longer sufficient to meet the demands that we just reviewed? Just a real quick one-slide primer on conventional toxicology, give you an example of what the challenges are with this. So if you take sort of an R&D or a product development pipeline, you can break it in many ways. Here's broken into sort of three phases: the R&D phase, the testing phase, where one gathers the necessary toxicology testing information to register that molecule to be sold in the marketplace. And then once it enters the marketplace, there's often not often, but occasionally, I should say, might be an issue raised about something that was missed during the toxicology testing phase. And so more toxicology testing is needed to address potential issues that may have been overlooked or the testing didn't pick it up in the first time around. So in sort of the conventional model is there's really minimal involvement of toxicologists in the early product design. The R&D scientists tend to work in silos, and they reach out to their toxicologists once they need that testing for registration. This approach it takes a lot of time, which is very important because you want to get a product in the marketplace as soon as possible. It does take a lot of money during that development because you're running a lot of different tests and using a significant number of animals in the conventional animal testing approaches. So just to give you an example, if you were to run 1-2 generation reproductive toxicity study, often done in rats, this uses over 3,000 animals to conduct those studies. It can run over $500,000 if things go well, and it's a very long time to take to run this. It takes over 15 months. Obviously, this is not going to work in a world where we have decreased timeline pressures to get products out on the market quicker. We have to assess more chemicals because of the different frameworks we're putting them through. So something else had to be developed, and that's really what we sometimes look at as predictive toxicology or new safety assessment. And that is, get the toxicologist into the R&D program as early as possible and help scientists develop less hazardous molecules. They work hand in glove, in tandem, to understand what parts of the molecule might be causing toxicity, and the developer needs to ensure that whatever parts of the molecule are needed for the functional capabilities of the molecule, the purpose it was being developed, are maintained. And that interaction can be achieved with the right approaches and the right test. By doing that, many better decisions are made. You can select a better, more sustainable molecule. This will also help understand what tests for conventional testing need to be run first. I should point out, predictive toxicology doesn't replace 100% conventional toxicology, but by using predictive toxicology, one can identify what the most likely hazard might be, and therefore that would be the first conventional study to run in order to determine if the prediction was accurate or not. So this is what we sometimes call informed testing or intelligent testing. We can use information for other non-animal approaches, such as read across, pick better formulations for molecules, and so forth. All right, if this is all done correctly, then this period of when the chemical's on the market can be reduced to nothing. You don't have to defend it anymore if something goes wrong because you get it right from the very beginning. Reduce that product development cycle. You also have more sustainable chemicals for consumer acceptance, but also sustainable in terms of the lifespan it's going to be on the market, and so recouping some of those investments. There's a very good business case to be made for green chemistry, especially in this complicated world of chemical management that we live in. Okay, so really predictive toxicology is the way in order to develop greener chemicals given these constraints and given these requirements. You can break it down in different ways. One of the ways to bend these is really into three sort of subdisciplines. One is using chem informatics, so computer software approaches, computer modeling, sometimes QSAR or quantitative structure-activity relationship. There are non-animal or in vitro approaches that can be applied for biological effects using cell culture assays, largely novel organisms such as zebrafish. and many high-density or high-content information gathering systemic approaches such as genomics, metabolomics, transcriptomics, things of that nature. So there's plenty of technologies that can be used to look at the molecular pathways that are affected by exposing chemicals to biological models in vitro. And then, of course, if one is going to take this all the way to safety assessment, need to do some exposure assessment as well. It's not just about hazard. Ultimately, we want to know what a chemical risk is, and therefore we need to know something about exposures that produce the hazards or the toxicity. Exposure modeling used to lag behind a bit. It's really catching up quite a bit, and to where now we can get some fairly accurate exposure predictions based on physical chemical properties. This is all combined or integrated in order to make a final prediction for safety. You have to pull it all together in the end. You always start with cheminformatics, then move into biological profiling, and then your exposure, and I'll lay that out again in just a moment. So that's the world, a complete world integrated approach to predictive toxicology, but one doesn't have to do everything. One could just focus, for example, on cheminformatics. And that's the approach that's really needed early in the earliest stages of R&D, where you don't even have chemicals to test in the earliest stages. So just to give you an example, there are plenty of cell culture assays available. Here's an example I took from a publication that looks at about 6 different assays that are available for skin sensitization prediction. This is cell culture largely, and it is looking at very key events in pathways we know that lead to skin allergy, skin sensitization. Each one of those can be queried. You pull 2 or 3 together, and if they are affected in the right pattern, the right order, then we make a prediction whether or not the chemical would be a sensitizer or not. And there are plenty of assays that are, sometimes we call them guideline studies, but these are validated assays that can be used to measure the potential for a chemical to produce effects for all kinds of toxicity, skin irritation, eye irritation, skin sensitization, genotoxicity, and so forth. That list is growing, and it's becoming a very important part of replacing animal studies, even in regulatory schemes, such as in the world of cosmetics, where some laws, such as the European Union, Australia now has brought this online, and many other countries are looking at complete bans for chemicals that are used in cosmetics that can't be tested in animals. And so really these approaches are the only requirement for many of those chemicals for new chemical development. So many QSAR cheminformatic methodologies have been developed. Again, cheminformatics is the integration of all the information on a chemical compound and its structure and its substructure analogs. So you can make a preliminary assessment of its potential toxicity or its potential physical properties or potential activity, even for drug development, for example. There are a lot of different ways to do this, but they can be bent into 3 major areas where there are statistical approaches. These are using often multilinear regression models, sometimes logarithmic regression models. You fit a training set of data using an algorithm that produces a linear regression. There are mechanistic models. These are predictions that are made using, again, a regression model, but now you're looking at very specific parts of the chemical features, toxicophores or structural alerts that are strongly associated with specific types of toxicity, such as gene toxicity, again, sensitization, or cytotoxicity. And then there are some nearest neighbor method. This is looking at the whole chemical structure, not just substructures. And here the predicted toxicity is estimated by looking at an average of a predefined number of chemicals in a training set that are most similar to the test chemical. And you look at the properties of those very similar chemicals around the test chemical. Very popular these days because of the different approaches and the multitude of models that are available is some sort of consensus method. And this is where an estimate is done by taking an average of the predicted toxicity from each one of these different QSAR methods above. And there are different ways to build consensus methods and predictions from taking the predictions of individual prediction models. So it's a lot more available, but in general, you can think of it in that way. So structure activity relationship tools are available that are freely downloadable or commercially available. This is just a short list. There's plenty more than this. The ones I've listed here are very commonly used in industry and in government. These SAR tools, though, don't work unless you have data. Fortunately, these days, there's plenty of data available publicly. And this is growing more and more because that earlier regulatory increased requirements for chemicals, all that data is, or much of it is now becoming publicly available. Once it's submitted to governments, parts of that information can be released publicly and be used to build cheminformatic models. If a company has its own proprietary data, that can be used also to add to the database to enhance the prediction for very specific chemical classes. Many chemical companies will focus on different classes of chemistries, and so they have some of their own tox data for that class of chemistry, and one can enhance these models by incorporating that as well. All this, again, has to be integrated and pulled in by an expert who understands the limitations and the strengths of each one of these models and each one of those different types of prediction approaches. There are strengths and weaknesses to each one of those. That is pulled together to provide guidance on whether or not a chemical is likely to be toxic or not for a particular endpoint. So a full-blown testing plan could look something like this. To use a non-animal strategy completely, one would start with a cheminformatic approach. QSARs are very commonly used, as I just mentioned. Our UL tool I'll talk a little bit about is called Reach Across. This predicts acute mammalian toxicities, as many other models are very good at the acute mammalian toxicity predictions. There's also environmental models that are available, such as FE-Suite Ecotox, for example, from EPA. If a hazard is predicted, one might want, and you have test material, that the chemist has made enough material to test, then you might be able to run some non-animal in vitro testing. This will have two objectives here. One is, first of all, to provide further information to confirm the cheminformatic prediction. This also provides a dose response, so one can get a sense on just how potent is the potential toxicity. You take a lot of chemical, is it weak, or is it a very potent chemical, and it takes a little bit to produce that effect. That dose response is very important ultimately when going into risk assessment. Okay. If you do want to move towards the risk assessment, this is important, then there are approaches to conduct what's called an in vitro to in vivo extrapolation, or IVIV. This is predicting what exposures would be required for a human, if it's human health, to produce that toxicity based on the results obtained from in vitro testing. And for more complicated biology, such as chronic exposure for developmental, repro, neurotoxic, chronic toxicity, cancer, and so forth, there are some specific mode of action-based studies that can be conducted in vitro, which provides some sense on whether or not you're likely to produce those kinds of toxicities. But in general, it's difficult to predict a lot of those more complicated toxicities from in vitro and cheminformatic alone. A lot more testing has to be done to cover enough of those modes of action. That information, though, again, can be folded into an IVIV so that if one can calculate potential human exposures, and even a risk assessment can be conducted, comparing the doses needed to produce toxicity with the doses that humans are exposed to, and you can get a margin of exposure to determine whether or not you're likely to produce toxicity in humans from the normal intended use of that chemical. So that's kind of a full-blown approach to how a pure predictive toxicology risk assessment might be conducted. You don't have to do all that. One can start just with cheminformatics, and that's what I want to talk about now. Just very briefly, you know, the world's changing because of all that toxicology data that's now available. That can be used to build models in ways we never were able to do this just 5 years ago. We only had maybe a couple of hundred, if we're lucky, chemical toxicity endpoints for any given endpoint we wanted to build a model for. You can't do much with just a few hundred endpoints. But when you start moving into tens of thousands endpoints, chemicals for endpoints, then you can start taking advantage of more modern machine learning approaches, artificial intelligence. And so there are databases now with some even with millions of chemical data points available. The top three there, ECHA, EPA, EFSA, are really growing quite a bit. Very important because these are regulatory submissions. ECHA's Reach database has over 20,000 compounds with multiple endpoints available. They're releasing that data that can be used for QSAR model development. EPA, over 2,000 chemicals they've released. EFSA, European Food Safety, over 10,000. And you can read the list, just going down. These are all publicly available now and can be used to build models and using any types of approach. What I want to talk about now is the approach that our collaborators at Johns Hopkins have used, and they have built a machine learning-based prediction model using structural similarity. And this, just to give you a real quick sense on on how these similarities are determined. If you look at these two chemicals, these are methacrylates. You have an isobutyl and a butyl. To you and I, these look very similar. To a computer, though, they have to be converted into some sort of code, a vector, a binary descriptor. And once that's done, the computer software can then begin to compare these two. Tom Lichtenfeld, who developed the software I'm going to talk about, he looked at about 8 or 10 different ways to do similarity comparisons, these different heuristic models. Fortunately, he landed on one of the simplest, that's the most widely used and widely accepted by regulatory agencies. This is called a Tanimoto index or heuristic, also called Jacquard. And it's very simple. It's just a ratio of the substructures that are in common, so the intersection divided by the total number of substructures for the two chemicals, so it's the union. All right, the intersection over the union, and you get a ratio. So these chemicals look similar according to Tanimoto. Those two are 90% similar based on the computer prediction. So it seems to be working. In fact, we know it works fairly well for most chemicals. Again, it's widely used. Using that approach then, what the software does is it builds networks of chemicals comparing the distance to one another using that Tanimoto score. So those lines or edges really represent distance based on that Tanimoto index. And here's a bunch of methacrylates that are very close together. This grouping of chemicals, these networks are called nodes. Build these nodes, and the prediction software produces a probability that a chemical is either positive or negative for a given endpoint based on the distance to either another positive chemical or another negative chemical, and which is based on their chemical similarity for the whole structure of the chemical, not just a particular toxicophora substructure. So just as a sort of a proof of principle, what Tom Lichtenfeld did is he downloaded 33,000 chemicals. These chemicals, we don't know anything about their toxicity. These are unknown toxic chemicals with unknown toxicity. And then he put into this mix chemicals that have known toxicity. He put in 300 and then ran the Tanimoto network similarity profile on them, and you start seeing the chemicals with unknown toxicity are associated with chemicals with known toxicity, forming these networks and nodes. And just by doubling that, almost every chemical now with unknown toxicity has mapped itself to a chemical with known toxicity. And then when he adds in the full number, he had 1,300 chemicals with known toxicity, every chemical found a very tight relationship based on chemical similarity with a chemical of known toxicity. So there was a really ability of chemicals with similar structures to tightly associate some with known toxicity and some without. That seemed, at least the networking part of this seemed to be pretty good. And so what he did then is he downloaded 10,000 chemicals from the ECHA database for REACH and built his networks. And now these black dots and these gray dots are these nodes of chemicals. So a lot of chemicals in each one of those. And they're forming these nodes of very similar structures. And they're forming sort of these supernodes. And then he layered on top of that, asked the question, how many of these chemicals are sensitizers? And so if they're blue, they are sensitizers, dark blue. If they're light blue, they're not sensitizers. And one can see now this very tight relationship between chemical similarity and sensitization and chemicals that are similar but also not sensitizers. So there's a real structure-activity relationship just seen from this pattern from these network of chemicals. This, I think, was published in Science, became kind of an iconic picture. So this was enough for you all to get interested. When we saw this, we said, all right, let's go talk to these guys. We like what they're doing. And long story short, we now have bought this software, and we have an ongoing career collaboration with them. We've asked them to validate these models using OECD principles for validation of QSAR models. He has built 9 models for prediction. These are all needed for this next round of REACH, fortunately, so they're available if anyone needs them. The number of chemicals, this is a little bit old, the list has grown, but we have a minimum of 2,000 chemicals now available for one of the models, acute quiet toxicity, but we have upwards of 15,000 chemicals for skin and eye irritation available for building predictions on. This is really unprecedented numbers for QSAR models. Usually, again, these are dozens, tens, maybe hundreds. We're into the thousands. And the analysis and networking shows that once you start around 1,200 data points or interactions possible, that's when machine learning starts to take a lead over just regression models. So we have plenty of data now to actually take advantage of the machine learning approach he's using. They're also fairly well balanced in positive chemicals and negative chemicals. and that's important in modeling, so you don't get biased results one way or the other. He ran this to determine how many were correctly identified the true positives, because we know their hazard classifications, and based on the thresholds we set for this study, at least 70% sensitivity, meaning true positives, were identified across at least 75% of the chemicals in that training set. So 75% of that 12,000, for example, for acute oral toxicity, were covered in this positive prediction here. So it's a very large, what we call domain of applicability. A lot of different chemistries are valid in this model. Many models, what you build them on really determines what domain of chemical applicability, what chemicals you can use that are valid to use in that model. And here we have a tremendous array of chemical structures because of the large data set we're using to build these models with. The balanced accuracy is the average between the true positives and the true negatives. Again, we can actually increase that, but we'd actually begin to reduce the size of that database because we're restricting the numbers of chemicals as you go up the specificity side. Anyhow, we wanted to see, we asked the question, well, how good is this relative to the gold standard, so to speak? With animal testing, Tom was able to pull out enough studies to do some statistics on the reproducibility of animal studies, and the balanced accuracies for those. They're pretty, they're not much higher actually for most endpoints than what we're seeing with our prediction model. So we're pretty comfortable that we're at a place where we're predicting with good accuracy the outcome of these toxicity endpoints that are very similar to the accuracy of the animal studies that we've been able to assess. So just to wrap it up then, make sure I give Pam a little time here. The chemoinformatics, QSARs mainly are really important for green chemistry because you can use them to assess human health and environmental endpoints, and they're quick, and that's important too, especially in the commercial world where you don't have weeks and months. You really want answers in days for your R&D team, so they're not waiting for your answer to make the next move. They have to be relatively inexpensive because you're going to be doing lots of them, and you can't really have a huge budget to test everything expensively. You need something that's quick and fast, and chemoinformatics really fits the bill there. Obviously, because you don't need test material either, you just need chemical structures. And you want to look for something that's user-friendly. There's a lot of different software out there. They're not always user-friendly, and that can take a lot of time to understand them. And sometimes you just don't have that kind of time to understand exactly all the nuances. There are a lot of resources available. I've just kind of pulled a couple here together where you can go and find multiple different QSAR models many are freely available, actually, like all the ones that are on the sites. You can also go to our site, Reach Across, and we can talk to you about how you can use our software without having to pay for it as well. So I'm going to stop there. And again, thank you, Amy. I love talking about this, and thanks for putting this presentation together.
Okay, that's wonderful. It shows that you like talking about this, so thank you so much. Pam, I'm going to hand over the presentation to you here. You should be receiving a notification on your screen there. And thank you so much, Craig. So we're gonna circle back to any questions after, but please do feel free to type any questions in the chat box there if you have them on your mind. Okay. Okay. I'll hand it over to you, Pam. Okay. Thanks so much. You see your screen.
All right. Great. Perfect. Well, thanks, Amy, and thanks, Craig. I mean, the first presentation is a great segue into what I'd like to spend the remainder of the time talking about, and that's really, where the chemists and the toxicologists work together, and I call that the crossroads of chemistry and toxicology, because that's really where the rubber meets the road to developing greener and safer products. So just briefly, I want to give you a little background about our products because it's important as we talk about the types of models and things that we're going to use as we're looking at product development. Some of the market challenges that we have, which Craig has already alluded to a bit, talk a little bit about our new product introduction and how we're integrating those safety screening strategies into that process, and that what some of our next steps are for actually expanding how we're using these tools internally. So just a little bit about our products. We are a company that is one of the only companies that produces nitroalkanes. So we're taking literally propane and ammonia and making nitroalkanes, alcohols, amino alcohols, and amino alcohol derivatives. So very unique chemistry. And these are used in a variety of applications because they have a variety of benefits to formulations as neutralizers, corrosion inhibitors, chemical, scavenging, many different applications. And so for that reason, you can see that the bulk of our markets that our materials are going into are things like the life sciences, paints, and personal care. So areas that we would say have broad-reaching exposure potential, reaching large populations of people. So it's very important as we're developing new chemistries in these areas that we're identifying early any potential EH&S red flags that we might have. As Craig alluded to, the challenges in these areas are really centered around ingredient safety. Are these ingredients safe? Do we know enough? How do we get more information? And then just ingredient bans that are happening, again, as Craig alluded to, when these ingredients are banned either by a regulatory agency or a big box company like Walmart, it forces companies that are making those ingredients to go and look at alternatives and how do they develop those alternatives. So I want to just talk a little bit about new product introduction and how does that work? What are we doing as we're looking at how we can find better replacements for products that are under pressure or under concern for safety? And I like to think of our new product introduction as this big circle that doesn't have an end. We're bringing all of the people that are from the important areas of the company together, commercial, product management, production, because we are manufacturing, our global supply chain, quality control, and R&D and sustainability, which I would consider, our area where our toxicologists work. And what I really want to focus on as part of the new product introduction is this relationship between sustainability and R&D, because this is really where we're focusing on new product development. The other things that happen from a regulatory perspective happen much later, but what we do here in the very beginning is very important as we're looking at new opportunities to develop new materials. And so when we look at what's happening with R&D and our sustainability teams, we're really talking about what's happening with our molecular design and synthesis, performance efficacy, and then how we're looking at the product safety profiles in conjunction with what the other part of the R&D teams are doing with the product efficacy and performance. Because what's really challenging in a lot of these areas is that when you're looking at a material that's going across a wide variety of applications, there's many different product formulations that has to be compatible with, and that does create challenges. So it's important to look at the efficacy and performance as well as what's happening from a safety profile standpoint. Craig showed this a little bit. I'm going to show it in a little bit different process, but this is really what we call our integration, how we're integrating the tools into our new product introduction process. And this is really just what we call a stage gate. And so very early on, when our project management teams come together, they're looking at candidates, and these are often things that they're thinking about before they've even synthesized an ounce of material on the bench. And as they start to look at these candidates, there are decisions that are made that are decisions that have to meet certain financial, safety, manufacturing hurdles for those materials to move on to the next stage in development. So really what I'm talking about today is this is the area in green where the chemists and the toxicologists work very, very closely together. By the time we get to this later stages of development, we're already ramping up for manufacturing, and to make decisions or design changes at this point is very difficult and it's very costly. And what we're doing with our early screens is really focusing not on everything, but just what we would call our toxicology red flags. And these would be considered CMRs, which are carcinogens, mutagens, reproductive developmental toxicants, and PBTs, those materials that are persistent, bioaccumulative, and toxic to the environment. Those would be things that would be showstoppers for us that if we were looking at a suite of potential new chemical options, we would rule those materials out for further product development. I want to talk a little bit about the importance of confirming the relevance of QSAR models for the target chemistry. Craig touched on this a little bit, but in our case, as I said, we have very unique portfolio of chemistry. Oftentimes, our chemistry is not included in the training sets of the QSAR models that we're using. And so we really have to do some upfront work to validate whether or not the models work for our chemistry or not. And so early on, we did do validation of a number of the different QSAR models that were available. We decided early on that TopCat QSAR model didn't work well for our chemistry. In some cases, there were models that we actually had to improve by acquiring additional data and enhancing those data sets, and that there were some models that just worked very well for some endpoints. So for example, genotoxicity, immunogenicity, many of the tools to a degree have very good reliable predictions for these endpoints. And then in other cases, we just had to agree that we needed to use a weight of evidence for some of those endpoints where we would run multiple QSAR models and look at that in the context of a weight of evidence approach. So what we did is we actually developed a flow chart for a screening level assessment where we have candidate chemicals now that come in, and we probably, I would say R&D and our group works together on a fairly regular basis where they're giving us anywhere from 5 to 25 different chemical structures that we now take through the screening process. And we have a hierarchy of the way we look at these materials, and we first look to see if there's any existing information. So we can search by name, synonym, CAS number, structure or substructures, and we can also go to databases to see if there are any chemical analogs that we could actually read across to existing data, make that assessment, and if that's enough, we can actually make some recommendations based on that data. If it's not enough, as Craig said, and many times with our chemistries, because they are so unique, there is not often any existing information we can use, then we have to go to the computational approaches. And so I highlight here the approaches that we're currently using for ecotoxicity, environmental fate, the programs, and the programs that we're using for our mammalian toxicology predictions. And then we can integrate this then into any additional information that we might have and report back to our R&D team what the next outcome might be. The outcome might be that we're good for now. We may propose that we may gather further information and testing later, or it may be that we need to go and do some additional testing right at that point in time. So let's look at how this really starts. And as I said, it all starts with the structure, and we're being asked to give an opinion. Which one is safer? Which one should we really choose to advance further on into our product development process and to invest further time and resources in? So as I said, we would look at step one, which is, to see if there are any chemically similar analogs. In this example, and this is just a fictitious example, but let's say we have a chemical that we see here. We find an analog that actually has some data on that. We can start to build a summary of what that data is and to see what that data tells us, and actually make some assumptions about the way this toxicity profile is for the chemical analog, how that might be read across to the existing structure for which we don't have any data, and we're trying to make that assessment. Where we have uncertainties in the data that we might gather, we can then go on and do further modeling. Here's an example where we could now model using the OECD toolbox for the candidate chemical for a variety of different endpoints. And I will say, with a lot of these QSAR models, these take some time and expertise to use. Oftentimes within the model for the endpoint, within the toolbox, there are multiple models for the same endpoint, and it's important to understand what you're running and what the output would be, but then we can then gather that information and summarize that. And again, we're just looking for alerts. Are we seeing anything with genotoxicity? Are we getting alerts that would be something significant that would be part of the red flag group that we talked about earlier? And then we would simply report out the safety assessment. So for the data set that we just looked at, there were really no significant alerts that resulted from that read across or modeling. And the OECD toolbox, the modeling results supported some of the information that we saw for the analog chemical, with the exception of skin sensitization. And then there were 3 endpoints that were actually out of scope in the model. So if we were to go back to that slide, and you can see that when we get inclusions rules not met, that means that our chemistry was out of the domain, and so the model would not be able to accurately predict the outcome for that. So in this case, there were 3 endpoints where the modeling was out of domain for skin irritation and corrosion and ecotoxicity. So we decided that in this case, if we were going to make a recommendation to R&D, it would be to move towards some in vitro testing to better characterize the endpoints that were out of scope for the modeling tools. And so in this case, the in vitro results, if we did go on and do that testing, the skin and eye irritation was non-irritating, and the skin sensitizer was shown that it was not a sensitizer. So we were able to increase our confidence in what we were seeing with the predictions from the available data that we had from the chemical analog, as well as from the modeling that was done. And biodegradation and ecotoxicity might also be a screen that we would move forward with in the future. But for this point in time, we would say, geez, we'll put that on hold because other factors have to come into the decision process. So we have to make sure manufacturing, the material is feasible, that the commercial opportunities are feasible. And these are all additional inputs that might choose us to make the decision to defer testing till we have other information that says the product development is really viable to pursue. So safety information is important, but it doesn't always drive the decision to go to the next stage gate in product development. So it's important that, you know, there's continued project management team engagement with the sustainability group so that as that new information becomes available, we can be ready to implement whatever those next recommendations would be for further testing, so that at that time we get to the commercial launch point where we actually have to conduct testing for registration and we actually have to perhaps run animal studies to meet those regulatory requirements, we can be sure that we're not going to have any undesirable health effects at that point in time or environmental effects. So just want to highlight some future work and to say that, you know, we do have a collaborative project underway with UL to actually validate the reach across tool for our Angus chemistry. We're working with Craig now to really outline a project and implement where we will repeat some of the modeling work that we did with our OECD toolbox validation to be able to determine if the reach across tool will have improved domain coverage and might actually be a better tool to use in place of some of the tools that we're currently utilizing. So this is kind of an iterative process as new tools come out, as we're looking at what those new tools can offer. We're evaluating them and we're trying to incorporate them into our new product development. So I'll just end and try to keep us on time and give us an opportunity for a few questions. But that certainly concerns over product safety can determine the success or failure of a product. We've seen that there are many instances that are external drivers that go beyond just regulatory drivers. But now companies can actually apply quick and inexpensive tools to estimate those EH&S issues early in product development. And so by doing that, we can actually save time and resources and hopefully bring more sustainable products to market. So we can highlight those concerns early, and then we can actually now have a more thoughtful approach to how we address those concerns with more targeted testing and more advanced evaluations, as Craig showed in his earlier presentation, that we can go from cheminformatics to bioprofiling to bringing in more mode of action type work in a very integrated and more intelligent test design than just checking the box and fulfilling data requirements as we often did in the past. And again, this just increases the company's ability then to launch sustainable new products in a more cost-effective manner, because what we want to do is have products that are going to have improved profiles, have longer product life cycles, and really reduce their impact to environmental and human health, but still work well. So with that, I'll end. And Amy, I don't know if we'll have time for a few questions, but thank you for this opportunity.
That's wonderful. Thank you so much, Pam and Craig. That was a wonderful presentation for giving us some really great ideas and for letting us know more about your work and what's happening in industry to consider this early in the design stage. This has been fantastic. I am going to let this go past the hour, but if anyone does have to jump off the webinar, this recording will be posted. So, and you'll actually receive an email tomorrow. So feel free to, if you have to jump off at the end of the hour here, feel free to come back and listen to the questions. So we do have a few that have come in, and if you have any outstanding questions, please do, type them right in there on your control box. We're gonna go through some of them right here, and I am actually going to just take the controls back from you, Pam, as well. But we have the first one that I'll read to you guys here is, are there some, this goes to some of our work here. Are there some design rules to be extracted from some of the QSAR models for chemists? For example, in the chemical similarity skin sensitization correlation example that Craig shared. So I guess, is there any sort of learnings or design rules that we can extract from some of those things?
Yeah, I guess I'll go ahead since you mentioned my name. I'll just tell you. Yeah, there are a whole catalog of what we call structural alerts that have been identified from the mechanistic aspects of what drives a chemical to interact with a macromolecule or cause oxidation or so forth. A lot of what chemists know about what makes a chemical reactive is relevant to it being toxic as well, because it is chemistry that's occurring in the cell as well. But those catalogs are available. They're called chemicophores, toxicophores, structural alerts. Some of the resources that I identified that will have backgrounds on that, there's some review articles as well. If you just search for toxicophore, when you're searching around there are a few toxicologists and scientists who really spend a lot of time cataloging that information. Many of the QSAR models have databases of those structural alerts, and that's what they use to query chemicals with to see determine whether or not do they have these toxicophores in them, and then the more they have, or the various combinations, we know there's a higher probability they'll have the kinds of reactions, the mechanistic drivers that would produce toxicity. So, an epoxide is always a bad thing in toxicology. We see epoxides, we get nervous and get heartburn. We don't like those. So, they're very reactive. They tend to do bad things. So, we could give you a whole catalog of those, and those catalogs actually do exist. So.
Yeah, and I would
Wonderful.
say, Amy, in working with Nick Anastas, he's sort of cataloged some of those, and we probably have those listed somewhere through the various workshops and things that we've done. Not a lot of halogens and all those kinds of design rules.
Great. Okay, we'll see if we can get those out to the participants too. That's great. I have another follow-up question here. Also, Craig mentioned free tools from Underwriter Laboratories on Reach Across. Can he please let us know what is available for use by educators?
Yeah, so we have a program we like to call our beta user program, where we are finding groups that want to use this, sometimes with companies, others with individuals, but we'll allow them free access to the tool in exchange just for feedback. And I can connect you with the right person to talk to about that if you want to just send me whoever is interested in that, and I'll be happy to connect you. Yeah.
Okay, perfect. All right, thank you. Okay. How often, this could be a big one, how often are the predictions aligned with the actual toxicological testing results, and what is the process for determining how a new chemical will be tested once being considered for development or entering the marketplace? I know you guys are doing, looks like, it sounds like, how often are they aligned with what you find early on?
So the validation work, I showed you the table, the different specific sensitivities, balanced accuracies. That's designed to give us a probability of how accurate the predictions are. So depending on how we cut thresholds off, we try and make a minimum of a 70% accurate We try to get closer to 80%, what we call sensitivity. All right, and then the negatives, the true negatives is the specificity. Those are the two key components you want to hone in on. We can do statistical testing to determine how accurate a model actually is. So, our evidence, because of the largeness of the database, we have a lot of chemicals now to compare against. That's improving the accuracies close to, at least in the assessments we've done, and getting close to what animals are reproducing themselves, the accuracies in animals. So if I take an animal, tested it with the same chemical against multiple animal studies, sometimes they're only 70% predicting what the results of the exact same chemical in the same species would actually produce. So what we're finding is, and that's the data we're basing these predictions and models on, so there's a limitation to how good it's ever going to be. And that's where the field actually is debating right now. Your question is very prescient, because that is the key question. How good do they have to be in order to rely upon? How much confidence do you have in them? Which means, what are we comparing them with? So it's a long-winded answer to say, depending on your model, you can have anywhere from flipping a coin to 70 to 80 to 90% accuracy for certain endpoints. The more accurate they are, you tend to limit the number of chemicals that are useful for, though. That is a trade-off often. So.
Yeah, and I guess I would say, maybe, Craig, just to build on your answer, I mean, the debate is right now, do we even compare test results to animals, especially as we get to the more complex endpoints? And I can say for our chemistry, at least for skin sensitization, using an animal test is not a good prediction of the true sensitization potential. And that's only been, realized through doing mode of action work that shows, because our materials tend to be highly irritating to the skin. There's been a lot of mode of action work that says that just irritation can stimulate a false positive in the animal, particularly the local lymph node assay that's used to assess skin sensitization. So in that case, we know that that animal model is not a good animal model for our chemistries, and we would choose not to use it. It probably a more predictive mode of action based QSAR would be better.
Interesting. Yeah, that's great. I got two more questions here for you. And our winner was Christine Jett at Toledo, so you'll be receiving an email from us. Congratulations. And how do you account for bias in the research that you draw your data from when making models?
The bias, okay, so there's two ways to do it. One is you use a test set of chemicals that were never used to build your models. That's one way, of course, you want to do that. And you can hold out chemicals, build a model with your set to build a model, and then your training set, and then you have a set of chemicals you do a test set on. There are lots of different statistical approaches to ensure you're not biased in your results. Also, the balance of the chemicals of positive and negatives is really important to make sure you're not biasing it. If you have a whole database built on chemicals that only produce toxicity, you're probably going to predict toxicity, right? You want to have both positive and negative outcomes in your data set. That's really important. So that balance of the data you're building, your training data, is very important. There's also challenges that are done. We're actually we're entering a challenge that the National Toxicology Program just put out there. And what they're doing is they're putting out training set chemicals they've very carefully selected. Everyone can use that same data set of training chemicals to build their model on. And then they're going to, you send them the model and they run it against a set of test chemicals that you don't know what they are, and then give you the results. So it's completely black box. You don't know. So there's ways to do this. That's a challenge, and we're entering that for acute toxicity. So the world's getting pretty clever on how we're going to do this, especially since everyone wants to build models now, and there's so much data out there that, it's really important to understand exactly what you're getting at. How do we know that we haven't tricked it into giving us great results, right? So that will become important to find, especially in the regulatory world, where we really need to ensure that we're getting the best and most accurate information. So.
Great. Okay, great. And our last question: which do you think is the most important driver of product EH&S concerns, global consumer demand or regulatory pressures? Which is changing more quickly? That could be a big discussion there.
Yeah.
Go ahead.
I mean, you need both.
I think you've covered this
but
well.
I'll say this because I'm no longer in a regulated industry, but I help a lot of companies who are in a regulated industry. I think the quicker driver and the more challenging driver are the sustainability metrics and the non-regulatory voluntary metrics, those frameworks, the screening, hazard screening frameworks. Those move quickly, and they get a lot of inertia behind them, when they get going. And chemical companies have to really informally have to respond to those. Those make decisions because you don't have the process that you normally go through for bringing, the time to bring in data and all these other things. Decisions are made, and your product's off the market. No one wants it. You can sell it, but no one wants to buy it, and especially at the speed of the internet, right? So that's a real challenging world. And but certainly regulatory drivers are going up. You can't avoid those either. So unfortunately, you need both for chemical companies, but they're getting better at understanding this now and really integrating that which is really the big driver for green chemistry, because you want to try and avoid all the problems you can because they're just so complex, right? But I think voluntary, maybe at least for the last 5 years, has been driving more decisions in chemical development than the regulated world. But that's changing because the regulated world's catching up now. So.
Yeah, I would agree, Craig. I mean, I think the regulatory world, at least we know what the requirements are and we know what we need to do. But on the voluntary side, that's a very volatile area, and we're not always, anticipating what's happening in that arena. And as you said, it's very fast. So no chance to, bring your case forward and have it evaluated.
Right. All right. Okay, well, that concludes our question and answer. So thank you so much, Pam and Craig. This has been wonderful. Thank you so much for sharing this with us and for all of your expertise in this space. And thanks everyone for tuning in to the webinar. Please check back on our website or sign up for our newsletter to be notified about our spring webinars. We have 3 in the works. We just hadn't nailed down the dates yet, so we didn't want to put up an announcement of our next one. So we have a couple months off, and we look forward to seeing you back here in early February. Have a wonderful holiday, and thank you again, Pam and Craig. Thanks so much.
Thank you. Enjoyed it. Bye-bye.
Bye.